Svante Pääbo was from an early age fascinated by ancient human history. While using DNA technology for his doctoral studies in immunology, he also attempted, as a side project, to use these modern tools for studies of ancient human remains. He soon realized that this is extremely challenging since ancient DNA is damaged by chemical modifications. It is also fragmented and present in extremely low amounts together with contamination from microorganisms and present-day humans. Svante Pääbo therefore embarked on a long series of rigorous technological developments that lasted over 3 decades. He performed much of this work on extinct animal species, but his goal was a seemingly impossible task. He aimed for our closest relative, the Neanderthal, that went extinct around 30,000 years ago. After setting new experimental standards in this challenging area, Pääbo first set out to retrieve and analyze Neanderthal mitochondrial DNA. He obtained a bone from the Neanderthal type specimen that had been discovered in 1856. From a small piece of this bone, he managed to extract and sequence mitochondrial DNA. He compared his new sequence to present-day humans and to chimpanzees and found that the new sequence was different from both of them. This was an important milestone. Pääbo had proved that it is possible to retrieve and sequence DNA from 40,000-year-old bones. Mitochondrial DNA, however, contains very limited information, and Pääbo's ultimate goal was to sequence the 3 billion base pair Neanderthal nuclear genome, which would allow in-depth analysis and comparisons across multiple genomic regions. The human reference genome had been sequenced in a huge international collaborative effort, and the chimpanzee reference genome sequence was also available. Sequencing the nuclear genome of an extinct hominin, however, seemed impossible. Pääbo embarked on his bold plan, and for this he needed more bone. Thanks to his strong international networks, he managed to obtain 3 bone specimens from Vindija Cave in Croatia, together with additional material from Germany, Russia, and Spain. All along, he refined and optimized his experimental procedures. By skillful use of new high-throughput sequencing technologies and by bringing together a large international group of collaborators for advanced computational analysis, Pääbo finally achieved the impossible, sequencing and assembly of the Neanderthal genome.